Hi everybody, welcome to this online revision session. I thought it'd be a good idea to upload a short presentation before the actual online or revision session that we're going to have in the next few days. So this is for the Molecular Genetics of Human Disease revision session with Dr. Mohammed Ahmed. As you know, this year has been quite a unique year. I wanted to bring up this image. Hopefully it looks familiar to most of you who attended the workshop for essay writing. And this is the template that we finished with at the end of our session. And the essay question that I wanted to tackle today was the following. Describe the RNA interference pathways and the effects of their mutation in relationship to human disease. So if this question comes up or a similar question comes up, or you should be very mindful of what the question is asking you. So the first thing is asking you is describe the RNA interference pathways. The second part of the question is effects of their mutations in relationship or in relation to human disease. So it's a two-part question in one. So you have to tackle it as is stated. So where is this information found? So if this question came up, all the information or majority of information will be found in my lecture titled RNA interference. So I'm just going to go through some of that today, obviously not the whole lecture, it's a short presentation, but just give you an idea where the information is coming from. So some background. RNAs that do not encode proteins, which are known as non-coding RNAs. Examples of this are tRNAs or transfer RNAs, ribosomal RNAs, small nuclear RNAs, also known as housekeeping genes. And the two key examples I want to talk about today are microRNAs and small interfering RNA molecules. In your essay to tackle this question in particular, you could actually go and mention any of these four that you want to do, but you need to mention at least two of these pathways. So I suggest I would focus on, or you should focus on, our small interfering RNAs and microRNAs and comparing those two in terms of RNA interference. But again, if you're interested in the PV pathway or the repeat associated pathways, you're welcome to do those as well. But today's session will be based on looking at siRNA and microRNA to answer this question. So what are siRNA? They are short interfering RNA molecules, 21 to 22 nucleotides, and they're derived from a long endogenous double-stranded RNA. You have microRNA, which are 21 to 25 nucleotides, and these are also endogenously transcribed, which I'm going to mention later on. You have the PV interacting RNA molecules, and you have the RAS, which is a repeat associated siRNAs. And the really important thing is that they all have similar effects mediated through a common pathway. They're not identical pathways, we have very key important pathways which you should be aware of for this essay. So a small RNA mediated gene silencing, so microRNA, small interfering RNA, PVs, and RAS siRNAs come under the umbrella of gene silencing, a small RNA intermediate gene regulation, also known as co-suppression, post-transcriptional gene regulation, or gene silencing, and it's collectively known as RNA silencing or RNA interference in what I have mentioned to you. These are two pathways that you could use for your essay in the description of your essay or explaining it. Remember, if you want to draw a diagram, it has to be hand-drawn or submitted part of the diagram, the essay answer, and it cannot be copied or pasted onto the document from the internet. This will not be accepted. So let's compare the two pathways. So you got microRNA on the left-hand side. You got the transcription of the non-coding RNA segment by RNA polymerase II, which produces about 70 nucleotide precursor sequence, which has this very characteristic step loop structure here. This is then exported to the cytosol, which is then processed further by another specialized, it's processed into, initially when it's transcribed by RNA polymerase, produces kind of long primary transcript of microRNA with the characteristic microRNA, is processed internally in the nucleus by a very specialized endonuclease enzyme known as dosha, which breaks it down into this very characteristic 17-nucleotide precursor microRNA. This is then exported to the cytosol. This is processed by another specialized endonuclease enzyme known as DICER, which produces the double duplex 21-25 nucleotide microRNA. Then this is incorporated into a very specialized complex known as microRNA RNA inducing silencing complex. When it's incorporated into that, it can translate inhibition, it can degrade stability of mRNA, and also can lead to heterochromatin activation. So basically, closing of DNA. Let's compare endogenous siRNA. So you can have exogenous siRNA, which is incorporation of small interfering RNAs to knock out or reduce expression of a gene of interest, which we scientists use quite a lot. So endogenous siRNA can be transcribed sense and antisense manner. They produce a RNA pol-1 derived transcript, so similar to microRNA. But the interesting thing you notice is that it actually doesn't have the dosha step, but then goes straight into the cytosol, processed by DICER itself. Then it produces a similar duplex complex of this microRNA, which is incorporated into a specialized RNA silencing complex. The same complex, just given a different name based on the siRNA in this context, and leads to mainly mRNA destability. And obviously that leads to mRNA translation inhibition. So these kind of marks here refer to inhibition, while the arrow refers to a positive impact. These lines refer to a negative impact, which is what microRNAs, siRNAs do. And if you're interested in the RAS siRNA, that's absolutely fine as well. And you can do that without any real issues or any problems at all. So it's up to you what you want to do in terms of the ability of that. So it's really up to you what you want to do in terms of which pathways you want to compare. So remember the second half of the question was talking about the effects of mutations of RNA interference on human disease. So this is the aspect you want to look at when answering that. So microRNAs act via interaction with target mRNAs. So mutations in the microRNAs leads to defect regulation of target mRNAs. So mutations in microRNAs may interfere with interactions, leading to gene regulation defects. The example I've given, just in this case, is microRNA 96, which mutations lead to hearing loss. But I'll also give you several examples. And of course, you can use your own examples you're interested in. It doesn't have to be these. But I do suggest to use at least 3 examples in terms of this question, if this comes up, of 3 different types of microRNAs or different types of non-coding RNAs, and then mutation leading to disease. Going back to the original image that I had at the beginning of our workshop, is that be aware that the essay question should be broken down into 3 key components. You have the introduction, and the introduction should define the RNA interference, so what is it by definition, describe the function and applications of RNAi, and what they are physically, so approximately 22 to 25 nucleotides long. The second part is the main aspect where you're going to answer the question, so main text. In this case, I've used microRNA and siRNA pathways. I wanted to draw them to make it easier for explaining, but you can write the pathway and the steps that are involved in that. And remember, this has to be hand-drawn and cannot be copied and pasted from the internet. And I can highlight the differences between the 2 different pathways. And part B of section 2, which is the main text, is answering the question, which is examples of different types of mutations for microRNA. Here I've given 3 examples already. You don't have to use these, but you can use others. I've used MIR29 for hearing loss, BIOL200 for epithelial mesenchymal transition. Has also used May21 in terms of skin cancer in my examples. But again, you don't have to use these examples, but I would expect 3 good examples of microRNA mutations and disease related. And the third section is conclusion, where you're going to use 2 to 4 sentences just to wrap up what you've been talking about in the main text, take home message, and just finish off with your kind of essay. And this is a really important aspect in terms of writing an essay, especially for me, is that you write the introduction and define the background for that. Second part is the main text, where you actually answer the question of the essay, which is made of what is RNA interference, which is partly answered in introduction, comparing 2 different pathways, RNAi, microRNA pathway, and siRNA, what the differences are, whether written differences or by diagram or combination of both. Has to be hand drawn, please. And the second part of the question is examples, so I'll give those examples as well. And then finishing off with a really nice conclusion of tying off everything together. So thank you very much for listening to me today. Hopefully this helps. I will meet you in the next session, and please have your questions ready, and anything you want to ask me, you are welcome to. So thank you.